Good evening, dear friends. We are continuing our very interesting topic, biotechnology. Last time, I quite remember that we have learned no information about genes. We talked about DNA, we talked about RNA, and then we understood what is a gene. Now, we are progressing in the subject. Today, I intend to tell you one process very detailedly so that will be the heart of genetic engineering. You need to know what are the sub processes involved in genetic engineering. So I need to, based on the information which is given already, using that information, I want to make you understand what is the process of genetic engineering. Genetic engineering is also called gene therapy. Subsequently, what all applications we are talking about gene therapy, like genetically modified crops, use in agriculture, use in various aspects, everything is dependent on understanding the process of genetic engineering or gene therapy. Now, before going to that, few words I am introducing. You learned up to gene. What is a genome? Genome is nothing but the complete set of genetic material available in one cell. That is called genome. I told you every human cell contains about 23 pairs of chromosomes, 23 pairs of chromosomes. That means 23 from mother, 23 from father. Put together 23 pairs of chromosomes will go into the offspring. That means every human being will have 46 chromosomes, but let us not talk about number 46. Let us know, keep in our mind 23 pairs of chromosomes. Every chromosome will have DNA. Every DNA will have lot of number of base sequences. DNA is in double helix forms. 4 nitrogenous bases form like steps in the ladder. It is a long chain. Any trait which is expressing outside is responsible for a gene. A gene can be tiniest part of DNA. So, what is the order of these genes forming a whole length of thread of DNA? There will be many genes, many nitrogenous bases are there. You can also call it a chain of nucleotides. Nucleotide means here sugar phosphate is the helical strand. That means phosphate. Phosphate molecule is attached to a ribose sugar or deoxyribose sugar. The phosphate of the helix, that means the side walls, and the ribose sugar or deoxyribose sugar is both are attached with a bond called phosphodiesterase. Then sugar is attached to a nitrogenous base. A nitrogenous base, a sugar, and then phosphate strand, these three put together called a nucleotide. That means in a helix, when two bases are attaching to each other, two are attaching to both sides, half of one nitrogen base and then sugar and then this strand is one nucleotide. Other side another base, another sugar, another phosphate is another nucleotide. That means no nucleotide chains. These are all. So here what happens, this DNA will not be in a continuous, as I am saying, as I have drawn the other day, double helical form. The formation will be like that, but all are no, they are distributed in the chromatin material inside the nucleolus like a loosely placed strands here and there. That means the pieces of DNA are strewn all around. They are not ideally, they are not no, in a clear form. Each piece will having these nucleotides, that means no, base material, base sequences, sugar molecules, and then phosphate. If you are taking all these pieces of these 23 pairs of chromosomes inside a cell, that means inside a nucleolus, of which the pieces are loosely distributed here and there, everything is brought in a sequential order, that is called genome sequencing. Genome sequencing is nothing but the total genetic material sequence in the order the nitrogen bases are all attached to each other. If you are bringing it to one line, all pieces are taken, they are all placed in a sequential order, so that you will be able to read continuously like alphabets or a book from one page to the end. If you are framing entire pieces in the order of these 23 pairs of chromosomes, that is called genome sequencing. Why this is required? You know, some nitrogen bases, 3 nitrogen bases, 2 nitrogen bases, you never know. The gene is nothing but a piece of DNA or a piece of some nitrogen bases. Unless you know every gene is responsible for some expression, some character, some expression for outside, that means the color of eye, the size of the eye, the size of the eyelid. Likewise, many characters, millions of characters would be there. Every gene is responsible for a character. In future, with gene therapy, you want to isolate a particular gene of a favorable character in order to be deposited into some other animal or plant through gene therapy. Unless you know which gene is responsible a particular character, you will not be able to isolate the gene properly. To know the sequence of genes, gene 1, gene 2, gene 3, gene 4, likewise, if you are arranging all genes in a sequential order from all 23 chromosome pairs, that means the genome sequencing is made by you, then only you can pinpointedly identify a particular gene, and you can cut that gene, you can take out, you can introduce in some other plant or animal for gene therapy. So for that purpose, initially genome sequencing will be done in the process, but here I am only asking you to understand what is a genome sequence. This carefully, one after the other, each strands, they will have a process to identify what strand belongs to which chromosome. Likewise, each chromosome, all distributed strands will be one after the other, they will be arranging, and then likewise they read the gene sequence of all 23 pairs of chromosomes. The entire arrangement of knowing where a particular gene is there in the chromosomes, this is called genome sequencing. Right. Now, everything it will be done manually. Sometimes it was. Now an advanced technique has come. The scientists call it next generation gene sequencing. In that, the machine will pick up gene portions, the machine will pick up the strands of DNA. Lot of number of strands will be there in the genetic material. Machine will pick up which strand belongs to which chromosome. Likewise, it automatically arranges all genetic material, all chromosomes, all genome sequencing in a scientific manner, systemic manner. This is a very cheap process, very fast process. Manual thing is not required. Everything is machine made. So this process now it's being used in gene therapy. I told about genome sequencing, and then generation genome sequencing. Right. We leave that there. Then how reproduction will be done? Okay. Any individual will have, suppose human beings will have 23 pairs of chromosomes. Mother will have 23 pairs. Father will have 23 pairs. Here reproduction, that means giving rise to offspring, that means next generation, is by two means. In plants and animals. In animals or plants, one method is sexual reproduction. That means there will be a mother and father, and children will be there because of the sexual operation, and then 23 pairs of chromosomes will come from mother, from the ovum. 23 pairs of chromosomes comes from the father, that is through sperm, and then fertilization, there will be in the zygote, which ultimately gives out to the offspring, will have 23 pairs of chromosomes. Then another method is asexual reproduction. There is no mother and father. From every plant, you can reproduce similar kind of offspring, and next plant can be produced, next bacteria can be produced, next fungi can be produced. These are all some of the method called asexual reproduction, where mother and father are not involved, either from any plant, male plant or female plant. So the next generation plants can be plants, bacteria, like that, they can be reproduced. But what are the advantages and disadvantages of sexual reproduction? Sexual reproduction, since mother will have a separate set of genes, chromosomes, father will have some another traits, both will combine together to form the child, means child will inherit some characters from the mother, some characters from the father. At the same time, there will be some characters which do not belong to either mother or father directly. That means a variation has come in certain other characters. If this character is a good character, you can introduce to some other area. If it is not a good character, then no, it is not good for that offspring. That means sexual reproduction will have positive characters when they are expressing, that's good. When some of the negative characters are expressing, that is bad, you can't take them out. But in asexual reproduction, what happens? Since there is no mother and father, what plant you are using, exactly similar to the plant which you have used for asexual reproduction, exactly similar plant can come out through asexual reproduction. There is no change in the traits. There is no variation. Exactly what plant you have used, similar plant will come out. This sometimes is called cloning also. Okay. This will have advantage because if it is a positive, entire positive features, everything gets expressed, and negative feature, you can eliminate it. That means no, you will use only the features of parent plant which are beneficial, then only you go for an asexual reproduction. But human, there is no asexual reproduction. A cell can give rise to, yes, any plant can be, there are two methods. SSP asked rose plant. Rose plant will have two mechanisms. One, Through sexual mechanism means no pollination. That means this flower, there will be male flowers, female flowers. Male flowers will have male flowers will have the pollen grains. Pollen grains are nothing but male material through air, wind, and birds. This pollen material, when bird is sitting on a rose plant, it carries the male pollen material with its wings. When it goes again it is sitting on a female rose plant. These pollen grains will get transmitted to female plant, male plant, female plant, their sexual reproduction, it gives out offspring. But asexual method is also possible in rose plant, where it can be, where no, in the research labs on its own, sometimes where male material is not available, then asexual reproduction, plant gives out to another plant. Grafting is another thing. Normally you see in nurseries where a plant part of some other material is grafted with the existing plant, so that no grafted part will give at a new plant. They take out and then no, that's the vegetative propagation. Correct. Manisha told correct. Manisha, you are correct. Good. You are understanding. So, asexual reproduction, sexual reproduction. Since we are talking about genetic material, we are now restricting to human. Vegetative propagation, we are not going. So, these two parts you understand. Now I am plunging into the complex mechanism called genetic engineering or gene therapy. Gene therapy is, genetic engineering is nothing but from a chromosome or from a DNA or in the gene sequence, any gene which is not required by us as far expression is concerned. That means that is not a favorable gene. You can take out, extract that favorable gene, and in that place you can insert unfavorable gene. Unfavorable gene, you are extracting a diseased gene, unfavorable gene from the DNA, and you are replacing with a favorable gene in the area where you have extracted the unfavorable gene. That means you are replacing a deleterious gene, a defective gene, a unfavorable gene with a healthy gene, a favorable gene. Then the gene is a continuation. Of course, here you have exchanged a particular gene, you have separated and then now inserted some gene. This insertion can happen from same species, it can be from some other species, but a favorable gene which expression we require, that will be introduced in the gene sequence, and that gene will be allowed to multiply many number of times, so that in the next generation this entire favorable sequence with the modified gene will get expressed in the favorable in the next generation. This is called gene therapy or genetic engineering. Suppose one particular aspect, let us say there is a mother and the expression is the entire sequence you require. A mother and father will have a black eye color, retina color, but you need green eye color to the offspring. You think that is a favorable thing. But when mother and father will have a black color, even the forefathers also do not have a green color, there is no possibility of getting green color coming into the next generation. So what do? In the genome sequence, you identify a gene which is responsible for expression of a black color of the eye. You carefully cut it and then you take it out. In that place, you take the another gene which expresses the green color from some other chromosome. Not from, it need not be from the same individual, it can be from anybody. You take that gene and insert in the place where black retina is to be expressed, then the entire DNA part is allowed to replicate. That means produce many number of times in the same way, so that when it is coming to the offspring, only green color will be expressed. This is called gene therapy. You can very well identify any disease you have. See, we have diabetics, and the diabetes will get automatically transferred to the next generation. Cancerous cells. These are all things now in terms of medicine. This is a very useful trait where you will be eliminating the possibility of transferring a disease from parent to the child. Likewise, the crops. Some crops will grow very tall, but it will not give sufficient yield. Some are very short, but their yield is very high. But here what is required? You need to allow the plant to grow tall, because that is a use for fodder. Cattle feed on the leaves. So that is also a possible plus character. But what for you grow any plant? This is for the yield. So our prime concern is to improve the yield from the plant. Fodder value is the secondary. Increasing the yield, that means what plant is giving more yield. The gene responsible for high yield will be introduced to this plant. Then automatically the next generation gives you very high yield. Likewise, when we are talking about genetically modified crops, probably tomorrow we will be dealing with all the crops. What is this? What all characters are introduced where? What crops are in operation in the world? All those things we'll be talking. Favorable genes getting introduced into unfavorable. Some of the unfavorable gene will be deleted. Favorable gene is introduced so that it is allowed to multiply. Then next generation will express favorable character. Here, what are the steps involved in gene therapy? First, from a cell, first step is to isolate DNA. DNA is the genetic material. From the chromosome, cell, chromosome to DNA. DNA you are isolating, first part. Then that means sequencing you would do, genome sequencing is ready. Then second part is you will identify, DNA is identified and DNA sequencing is done, second part. Third is you will identify a gene of expression of your desired expression, identifying a gene part from the long chain of DNA, where from up to what point is this particular gene. You will have to identify. That means identifying the gene is the third part. This identification of gene is called gene of interest. You have certain interest in that gene. That is called gene of interest. So identifying gene of interest is the third part. The fourth part, you will cut the gene of interest, both portions, that side, this side, and you will isolate the gene. Cutting the gene of interest part is the fourth step. Then next step is after cutting the gene of interest, you will have to make it multiply. Here the mechanism, I will tell you how this gene of interest will become an mRNA, I will tell you. But you simply understand the steps. Remember the steps. We have cut the gene. This cutting will have certain enzymes will work for cutting of the gene. I will come to when I am explaining each step now, I will tell you. After that, you will be allowing the cut part replicate more number of times. That means you are integrating this gene. It will be transferred to some another DNA, a carrier DNA. A carrier DNA makes this particular part integrate with its DNA, and then it starts replicating many more number of that particular DNA. Then DNA becomes the fully operational, because your introduced part becomes automatically, it is replicating many more times. The last step is this replicated new DNA will get integrated with host DNA. That means where you are introducing this particular gene, where you are particularly introducing, where you are again replacing, where replacement from the original DNA, from one DNA you have done it, from the second DNA where you are introducing this favorable trait, there also you are introducing this new thing, and so that it becomes naturalized with the existing DNA of the host. These 5 steps are involved in gene therapy. Now I am going with each stage. First stage is isolating the DNA from the cell. Through a technique, you will elute the DNA. You will that DNA portion you will identify. The second is, I told you how gene sequencing is made. You will use the automation mechanism, and then now genome sequencing is done. Then third part, area on the DNA strand need to be cut in order to take out the favorable gene. Here, one thing you need to remember, and 2-3 times this has been asked in civils over the past 7 years. For cutting of the DNA, it means now you can call it a DNA scissors, but it's only for our colloquial thing we have named it like that. Otherwise, this is called an enzyme called CRISP, CRISP capital R, and CAS9. CRISPR-Cas9 is an enzyme which cuts the DNA to take out the favorable gene. CRISPR-Cas9 is the enzyme. You remember that. Likewise, another enzyme is there, E. coli, ECO capital R small I. E. coli is another enzyme. This also used for cutting the gene, cutting the DNA. These enzymes are called nucleases. That means simply the categorization of enzymes. This enzyme is called a nuclease. Then this enzyme is called a restriction enzyme. Their nature is nuclease. They are categorized into nuclease. But this CRISPR-Cas9, CRISPR-Cas9, and then E. coli, such kind of an enzymes which are used to cut the DNA to extract a particular gene is called, these enzymes are called restriction enzymes. This is another word which you need to remember, restriction enzymes. Restriction enzymes, they are not common for every cutting of the gene, but they are specific. One particular gene, if you want to cut one particular enzyme, one type of enzyme can be used. These kind of enzymes can be many, but in a current technique, 3 or 4 enzymes are being used, and they are very specific. One enzyme, suppose CRISPR-Cas9 cannot be used for every gene removal. It can be used for certain kinds of genes only. First thing, these are called restriction enzymes. Third part is where it cuts. It cuts not everywhere. It cuts only from the phosphor to sugar bond. That is the area where enzyme cuts the DNA. it eats out the DNA there, and then here this strand, there another area also, similarly there it will be cut. In between DNA part will be taken out. Well, cutting of the DNA, I told you. Then what happens to cut portion of the DNA? That means no gene, what happens? It's nothing but it's a nitrogenous base sequence. It can be 3, it can be 4, any length of that. It is called a gene, otherwise you remember as nitrogenous sequence. Well, how, whether the cut portion, if it is remaining like that, if it is not getting expressed in the host where you are introducing, if it is not getting expressed, then what is the use of cutting DNA? You should have some mechanism where you can make this piece come live and produce similar kind of sequences, no, many more. Then only it becomes expression in the next generation. For this, automatically this piece which you have cut from the DNA. You have taken it out, and that becomes that left a gap there. You have to replace that with a favorable gene. Okay, somewhere when you have already identified, I told you how to cut this DNA. Similarly, where you want to cut the favorable piece, there also similar mechanism. You are taking out that particular DNA, a gene. You have to introduce that gene, favorable gene, in the gap which you have created by cutting this existing DNA. If you place that portion here, can it automatically replicate many number of times? No, it cannot. You will need a carrier, that means a helicopter like thing, a carrier in between. Something has to carry this gene on its body or on its DNA, and after that it gets regulated into that DNA. It becomes naturalized and it produces many more strands of the DNA. Then only you can introduce into the host. For this, plasmid is the cell which you use for this mechanism. These carriers are called plasmids generally. Sometimes a single-celled bacteria will also do. Sometimes E. coli, Escherichia coli is a bacteria. This also acts as a carrier. Plasmid, E. coli, or some of the bacteria will act as a carriers. E. coli, I told you, E. CO, capital R, small I, is an enzyme which is used to cut the gene. E. coli is a bacteria. It's a good bacteria which is there in a human body. This bacteria can also be used as a carrier. What happens? A plasmid is nothing but a single cell having only one chromosome. Plasmid is another organism. It's nothing to do with the human body. You will introduce this particular gene portion which is cut into the DNA of this plasmid. Then plasmid DNA, that means now you are creating a space in the plasmid DNA. You are introducing this particular gene into the plasmid DNA. Then the gene of interest, that means now the portion which you have cut and introduced in the plasmid, plus already existing plasmid DNA, both together is known as, that genetic material is called mRNA, messenger RNA. Why it is called messenger? Plasmid is a messenger carrying your good gene to the host. That means there the cut bit joins the plasmid DNA. It becomes a recombinant DNA or DNA. Recombinant DNA. Recombinant means it will reproduce many times. It's a new combination, produces many more DNA with the favorable host gene. Then the recombinant DNA will be introduced into the host DNA, where you have cut for taking out the bad part. There you are introducing the recombinant DNA to produce many more number of this favorable trait. Automatically, this favorable trait, no, when it's going on for the multiplication, automatically that particular gene, when you are no new genetic material is formed, then only favorable genetic material will be passed on. Likewise, it gets expressed in the next stage. Recombinant DNA is nothing but the favorable part which you have added to the carrier, that means plasmid DNA. Both part together is called recombinant DNA. The recombinant DNA is again introduced into the host, where you want to introduce no good